Hello everyone. Today we present pseudo-segmentation guided GANs for brain tissue in painting for the BraTS in painting competition. Our team is Junyoung Ha, Jong-sung Park, and Ji-young Oh. We propose an anatomy guided 3D GAN for brain in painting with these three parts: a generator, a discriminator, and a segmentator. For generator, it fills masked brain regions with the healthy mask and tumor mask. For discriminator, it encourages the realism through an adversarial loss. And segmentator, it adds structural guidance through mask-aware perceptual loss based on pseudo-whole brain segmentation. We use different pipelines for development and inference here. For development, inputs are T1 MRI, healthy mask, and tumor mask. We create pseudo whole brain segmentation with SynSeg here. The model input has 3 channels, which is voided T1, healthy mask, and tumor mask. We train on 128 voxel patches and normalize intensities from -1 to 1. And for inference, we're given T1 plus 1 mask. Here we treat that mask as healthy, set the tumor channel to zeros, and again feed the 3 channels to the model. Here, training uses 3 UNet-based network. For generator, it takes the 3-channel input and inpaints missing region to be visually realistic and anatomically plausible. And for discriminator, it learns to separate reconstructed voxels from inpainted voxels. Here, reconstructed areas act as a realism reference, and the adversarial loss nudges the generator towards the realistic output. And segmentator, here it trains the pseudo-annotation. We compute a perceptual loss for the generator, which means it features space differences between the original and inpainted voxels, guiding the structure. For loss formulation, training brings 3 parts together. First, the discriminator, it learns with binary cross-entropy, labeling voxel as inpainted or reconstructed. And the segmentator, it's supervised with voxel-wise cross-entropy to the pseudo-labels, so its features reflect valid anatomy here. And the generator, it balances total 4 goals, which is a reconstruction L1 loss, a mask L1 loss on mask region, an adversarial loss from discriminator, and a perceptual distance in the segmentator's feature space. So these forces push the model to look realistic while staying anatomically consistent too. On the quantitative side, using MSE, PSNR, and SSIM, our model trails the current SOTA by a moderate margin. Qualitatively, the results look stronger. Here, the top row is development set. The blue outlines, as you see, are the healthy mask, and red outlines are the tumor mask. We use the blue region to judge the reconstruction and compare them to the reference next to it. In the red regions, we could look for tumor areas being filled with the healthy-looking tissue. We could notice the smooth boundaries at mask edges, continuous texture, and consistent tissue contrast. And the bottom row is validation set. As there's no reference inside the voided areas, so we judged instead the plausibility and could found a coherent anatomy, minimal seams, and balanced intensities across the tissues. So overall, the visuals are realistic and anatomically reasonable, which complements the qualitative results. Thank you for listening. If you have any questions about the presentation of our method, please email us.